Hello everyone, this is Dr. Rajni. Today we will discuss about the rDNA technology. What is the rDNA technology? How many objects are involvement is present? The first one is the how to use the probes, restriction enzyme map, gene cloning, gene library, cloning vectors, DNA fingerprints, and how to footprint of the DNA. Last one is a genomic mapping. These are the objective for the DNA technology. Coming to the DNA technology, here main important three techniques involvement is there. These are the facilitate and analysis of the human and animal DNA. The first technique is the restriction endonucleases enzyme. The second one is how to clone the DNA. Third one is the probes. In the coming to the first one, restriction enzymes cleave DNA into the smaller, more manageable fragments are present. Coming to the clone, how to clone the DNA. Here when they cut the DNA, that cutting name is called the DNA fragments. That DNA fragments must be amplified and to be more useful for the cloning vectors. Coming to the third one, here third is the probes. After DNA fragments, a specific fragments can be identified using a complementary probes. The each and every step utilizing the genetic engineering complete the rDNA technology. First step is how to cut the DNA. After cutting the DNA, you can take the one specific site. Cutting completion of the cutting, joining of two DNA fragments to create a novel DNA. Cloning or amplification of available in DNA technology. This is the expression of a DNA obtained from the product, and sequence of DNA molecule synthesis and analog nucleotides also involvement is the each and every step of the genetic engineering. The application of rDNA technology, first understanding the disease, that is for example sickle cell anemia, thalassemia, like that. How to identify the diagnostic disease, that is called the AIDS, hepatitis, like that. How to treatment. In case first you can take the human insulin, that is a treatment of the diseases for the rDNA technology. Prevention is called the hepatitis vaccine is the prevention. And what is the therapy? SCID is the therapy. Now coming to the DNA technology, how to vaccination preparation, how to gene cloning, and what is the gene jumping. These are the utilizing for the in this recombinant DNA technology. Two DNA fragments of insert, even from different source or different species, coincident or strictly ends with the complementary sequence, and in some cases blunt ends are joined by the homologous tiling. A small synthetic duplex oligonucleotides also having restriction site of attachment. Here DNA cut always 5' end to 3' end or 3' end to 5' end. 5' end of the linker DNA phosphorylated by phosphonucleate kinases also involvement is present, and this is a different enzymes utilize the DNA technology. First enzyme, restriction endonuclease enzymes, polymerase enzymes is the most useful for the enzymes. DNA ligase enzyme functioning is the joining the end the DNA molecules. Here another one is the polymerase enzymes. Here polymerases also three type, poly 1, poly 2, poly 3 enzymes are there. Poly 1 enzyme, the function of poly 1 enzyme, synthesis of the double stranded DNA. Another one, poly 2, this is the production of the nix of the sDNA molecule. Coming to the third enzyme, exonuclease enzyme, removal of the from 3' end of the DNA. Exonuclease enzyme remove the 5' end, polynucleotide kinase phosphorylate 5' end, alkali phosphates remove the 5' phosphate, S1 nuclease degradation of the sDNA. These are the enzymes are utilizing for the rDNA technology. Here rDNA technology recognition sequence of restriction endonuclease, E. coli bacteria, here 5' to 3', 3 prime to 5 prime. When read the 5 prime to 3 prime direction, the sequence on the top strand is identical to the bottom strand, that is called the 5 to 3, 3 to 5. Both are the recognition and sequestration of the restriction endonuclease enzymes. Here next coming to the nomenclature of restriction endonuclease. Nomenclature endonuclease is the 1, 2 and why the name to, so these are the strictly end of DNA or cut, blunt cut of the DNA, and restriction sites also frequency are present in this site. Here restriction endonuclease, how to the cut, the 4 bases is the quiescent needs are present, that is the restriction side or blunt size. What is the mapping of the restriction? Involves the size analysis, restriction fragments produced by the several restriction enzymes individually and in combination time. Another coming to the treatment, here different enzymes are there and measurement sizes are there, interruptions are present. Enzyme A, enzyme B, enzyme A plus B both are the involvement is there. The measurement is the 9 kb fragments are there, the interruptions are present. B is the 3 plus 6, either 5 prime to 3 prime, 3 prime to 5 prime. These are the measurements of the enzymes. What are the probes? Probes meaning 15 to 20 long oligant used to the search a particular rDNA fragment. These are called the probes. Here probes also chemically synthesized DNA or as usual RNA pieces also. Generally here labeled with the radioactive material or fluorescent label. When the probes you can use that one, in that time you can labeled from the radioactive material or fluorescent labels also. Next one, how to construction of the probes. From a genetic database, homologous gene other species or mRNA. Protein rich trap to MET, tryptophan to methionine probes are there. How to labeling the probes? The end labeling 32 probe or random labeling during synthesis of the DNA utilizing to the GTP fluorescent labeled is the present in the probes. The end labeling of a gene probe, 5 prime end and alkaline is the phosphate and polynucleotide kinases are there. In case the 5 prime to 3 prime end are there, this is a purifying gene probe fragment or synthesis oligonucleotides. Alkaline phosphates treatment or probe to remove for the 5 prime phosphate. Here polynucleotide kinase transfer phosphate group from donor 2 meaning 5 prime end to 3 prime end, that is the probe. 5 prime end of probe is radio labeled and gene probe is purified. The end labeling of genes probe 3 prime end is using terminal transfer system. Here the first 5 prime to 3 prime synthesis oligonucleotides are purified gene probe fragments. Here transfer the label DNA-TP to 3 prime end using terminal transferase. 3 prime end probe is radio labeled and gene probe is purified in the end labeling of the gene probe. Coming to the another cloning system. Here clones meaning vehicles or vectors. In the rDNA technology, very useful for the clones. After preparation of vaccines or genetical modification of plants, anyone, the clones is the must. That's why cloning character is the in the DNA technology very important. Here clones different types of clones are there. The process of producing a large number of identical copies from one single original DNA molecule or one DNA fragment to produce to the one single to large copies to produced in this cloning method. Importance of cloning. Why important? Here to study purified from the gene and insufficient amount also present to study the sequence of DNA, expressions and different tissue under the different conditions. Here methods, what is the in the cloning methods, amplifications of cell based method 1 and cell free method 2 are there. See, this is the total outline of gene cloning. First you can observe the picture. The first one is the one DNA fragment. This DNA, you can take the best example for the E. coli bacteria. The E. coli bacteria DNA, you can take E. coli bacteria and cut DNA containing enzyme, desired enzyme. What is the enzyme? Restriction endonuclease enzyme, first to cut the sequence of the DNA. The second one, plasmid. What is a plasmid? Both are combination is called the recombinant. Yes or no? These cut plasmid to insert the cut the DNA. Here cutting is the utilizing the enzyme endonuclease enzyme. The plasmids is different types of plasmids are there. PUBC, plasmids, cosmids, vectors, like this. Any one plasmid you can take, and both cutting of the DNA, plasmid, both to the combine with together. Here to attach the DNA, this cutting DNA and plasmid utilizing the enzyme name is called ligases. This is called the recombinant plasmid. The total structure is the recombinant plasmid. After plasmid, recombinant to complete the recombinant plasmid, this is to now to transfer to the bacteria. Another, this is a new type of the plasmid, cutting the bacteria and plasmid both are combined with the recombinant plasmid. This recombinant plasmid now to transfer the another bacteria. What happened? Another one, the cells are grow and select the after growing of the cells, you can select the recombinant colonies only. In the industrial, in the vectors or in the laboratory, after developing of the bacteria, in that bacteria you can select only recombinant colonies only. That is called the select colonies containing, when the you can after selection completion containing desired genes are you can insert that one. After desired gene, grow cells to obtain required quantities of genes are present. This is the total outline of the gene cloning or recombinant DNA. See here each and every step, how to isolation of, how to status of gene bank. Gene bank meaning just like the gene library is called the gene banks. Each and every vector containing a different foreign DNA fragments. So in the picture, two are there, one is a light color is there, another one is a dark. This is the light color is there, in that one another bacteria DNA, and dark color part is called the plasmid. After in the total select of the gene bank meaning gene library, each and every vector to contain different foreign DNA fragments. After cutting, next one, isolation of gene that is from the library, gene library screening method. The second step is called the screening method. After screening method, amplifying single clone from gene library from future and further analysis also important in the DNA technology. What is the meaning of gene library? Here the gene library meaning all of the DNA extracted from an organism and designed with a restriction enzyme will result in a collection of the clones. This collection of clone is known as called the gene library. What are the library types? In the gene library, first one is the genomic library. Genomic library meaning the total chromosomal of DNA organism. This is the genomic library. What is the cDNA library is there? Represent the mRNA from a cell or a tissues act as at a specific point of time. This is called the cDNA library. The types of gene library depends on final application of the DNA. What is the goal? Main important, production of a new or modified proteins or determination of tissues, specific expressions and timing patterns of the cDNA library. This is the goal. Another goal, to understand the control of protein production for a particular gene. This is called the genomic library. Another one, in the genomic library, the first step, construction of gene library. This is the digesting genome, DNA molecule, choice of the enzyme, any enzyme to choice, and the second one, ligation, to attachment of the DNA molecules. Here carried out by the 100C to lower than kinetic energy utilized, and the reduced chance of the stick ends of the patterning. And the next one, cloning vectors, and the screening of the gene library is the utilized in this one. See here in the table to represent the number of clones required for the rDNA technology in a genomic library. For example, the bacteria, best example for the E. coli bacteria. In the E. coli bacteria, the genome size kb, 4000 kb, and how many clones are required? Here in the 4000 kb, the fragments is the 17 kb fragments, 35 kb fragments also utilized. In the 17 kb fragments, E. coli bacteria fragments is the clones are the 700, 35 kb 340 fragments are there. Just like another microorganism, we can take the yeast. Yeast, the genomic size is the 20,000, the 17 kb fragments is the 3,500, 35 kb fragments is 1,700. Coming to the fruit fly and man, maize, these are the clones required for representation of the genomic library. This is the fragment produced by the cleavage with BAMHI. Here ligation molecule, DNA, how to ligase the molecules. Ligation molecules with coincident ends, complementary ends base pairs, forming a temporary link between the two DNA fragments. This association of fragments is stabled by the formation of 3' to 5' phosphodiesters linkage between the coincident ends. A reaction catalyzed by the DNA ligases are present. This picture comparison from the general step in the construction of genomic DNA and cDNA library. Genomic library, here cDNA library, genomic library, the first step, extract the chromosomal DNA. After next step, digest chromosomal DNA with restriction endonuclease DNA, endonucleases. Next, coming to the insert each DNA fragments into vector, that is vector is the recombinant DNA, and after the completion of the recombination, to transform the bacteria, clone each recombinant. This is the genomic library. What is the cDNA library? Both are different. In the cDNA library, extract the mRNA and produce the cDNA. Insert the each cDNA into vectors, that is a recombinant DNA. Transform bacteria, clone each and every recombinant. This is the difference in between, not difference for the both are the general steps for the each and every step. What is the next? Coming to the cloning vectors. Cloning vectors, DNA elements that may be stable, maintained and propagated in a host organisms, for which the vector has replication functions. Next, host organism is a bacteria. Which bacteria? For example, E. coli. In the total DNA technology, best example for this E. coli bacteria only. The E. coli vectors with a replication or origin in E. coli will replicate together with an incorporated DNA efficiency, and comparison of vector generally available for cloning. The vector is the M13 vector, host cell is the E. coli, vector structure is the circular virus, insert is the 1 to 4 kb, that is a range. And second vector, plasmid vector. Plasmid also host cell E. coli. What is the vector structure? Circular plasmid, 1 to 5 range kb is present. Coming to the third vector, phagase lambda. Phagase lambda also E. coli host, linear virus, 2 to 25 kb range are present. Next, cosmids. Cosmids also E. coli, circular plasmid, 35 to 45 kb is present in such range. And BACS, E. coli, circular plasmid, 50 to 300 kb. Last one, East YACS. This is the vector S. cerevisiae. This is a fungi, linear chromosome, 100 to 2000 range of the kb is present. See, this is a picture, you can observe to the picture, PBR 322 plasmid cloning vector. This is the structure of E. coli bacteria. In the E. coli plasmid, the cloning vector is PBR 322. Here different colors are there, no? This is the ampicillin resistant, where the orange color reflect is there, ampicillin resistant, and where the pink color is there, that is the tetracycline, tetramycin, the like this. The green color is the ori DNA. The E. coli and ori DNA is there, restriction for the tetramycin, tetracycline antibiotics, ampicillin antibiotics, and original DNA. This is the total plasmid cloning of the vector. That vector name is called the PBR 322 vectors. Coming to the picture, replica planting to the detect recombinant plasmid. Here this is the velvet plant, and in this chart, ampicillin plate with clones. These clones replicate to transfer from the replicate. After replicate, this is a tetracycline plate. Tetracycline plate incubate the only cell with plasmid, but without insert grow is there. This is total recover colonies containing recombinant plasmid from the ampicillin plate. This is the map and important features of the PUC vector. Another vector name is called PUC18. This also ampicillin resistant gene is present. LAGZ beta galactoside genes also present, and in this total vector name is called PUC18. Here kb is called base pairs, 2686 base pairs involvement is there. Ori meaning origin of replication. LACL is the control recognition for LAGZ, and these are multiply cloning site polylinkers also present into the PUC18. Coming to the principles of blue or white selection for the detection of recombinant vectors. After completion of the cloning, two colors of the colonies to form. One is the blue color colonies, the second one is the white color colonies. In this one, some are the non-recombinant vectors are there, some are recombinant vectors. Where the non-recombinant vectors are there, that is a no insert. Where the recombinant vectors are there, that is the insert within the MCS. Non-recombinant vectors, that is the induced with the PTG, that is the beta galactoside gene, to form to the X-gal hydrolyzed with white to blue color. This is the blue color points are there, and this is all the no insert. Where the another recombinant vector is there, that is the insert with the MCS. Induced the IPTG, here DNA insert in MCS, beta galactoside gene, X-gal not hydrolyzed. Where the not hydrolyzed are there, that is a white. that is a white color plate is the visible. These two, that is a blue color and white color and non-recombinant vector, so coming to the recombinant vector, both are present. Coming to the what is the cosmid, different vectors are there, no. Coming to the plasmid is completed, another vector name is called the cosmid. Cosmid vectors incorporate cosist from the phage DNA or phase 1 and also the essential features of a plasmid, such as a plasmid origin of replication, a gene drug resistance and several unique restriction site for a insertion of the DNA to be cloned. This is the antibiotic resistant gene. Which one? Cosmid is the antibiotic resistant gene. This is the cause site and restriction site, both are present. How to express the vectors? Okay, different types of vectors are there. These vectors, how to express? Insertion of a strong promoter and strong termination codons also present, adjacent distance between the promoter and cloned gene, the insertion of transcription transmination sequence are there, the insertion of a strong translation initiation sequence also present. The inserted sequence must be placed so that it is reading frame is in phase with the regulatory sequence. And the another one, shuttle vectors. The shuttle vectors have origin from replication for yeast and bacteria, both are there. Bacteria, you know that already has read out, no, E. coli bacteria. Another one microorganism, that is the yeast. Both are the shuttle vectors. This means that constructs may be prepared rapidly in the bacteria and believed into a yeast from expression studies are there. This is the shuttle vectors. And in delivery of the vectors into eukaryotes, okay, in the all are the prokaryotes is complete. Now coming to the eukaryotes. Eukaryotes is the transfections or recombinant molecules into animals, making the first step, making the membrane permeable with divalent and electrophoresis method, that is subjected to pulses a high voltage gradient, and liposuction. DNA is encapsulated by a core of lipid coat particles. This is the eukaryotic vectors. Coming to the screening gene library, the colony hybridization techniques for locating specific bacterial clones harboring recombinant plasmid vectors containing to the desired DNA fragments. Coming to the how to applications are present in the gene cloning method. Molecular analysis of the disease, normal gene variations are there, that is the polymorphism, and gene variation causing disease, beta globin gene, detection of point mutation, sickle cell diseases, detection of the deletions, insertions, rearrangement and beta globin gene. This is the pentalar diagnosis and IVF, disease linked analysis, microcetyl repeats, families forensic medicines. These are the applicable for the gene cloning method. This table, to discuss about this table, the total structural alternation of the global gene, that is the point mutation, deletions and the arrangement also present in this one. Here point 1 mutations is called the sickle cell diseases, and this is the schematic positions are there. The sickle cell disease is like this, all are there, and pedigree analysis also present in this one. These are the DNA required, and another DNA finger means, see here picture, the gene to how to parent to mother and children, so you can observe the these are the DNA fingerprints are the present in this one. What is the purpose of DNA? Paternity disputes and criminal identifications are used for the this one. Methods is these are isolation DNA, design DNA, amplification, bloating, all are the autoradiography. These all are the DNA fingerprints are the present, and DNA footprinting is the enable, control and reason to be position of the footprint of this one. This is the importance and recombinant DNA technology used for the many techniques and utilizing for the vaccines, antibiotics. These are the use for the technology. Thank you.